I'm Dr. Nathaniel Chin, and you're listening to Dementia Matters, a podcast about Alzheimer's disease. Dementia Matters is a production of the Wisconsin Alzheimer's Disease Research Center. Our goal is to educate listeners on the latest news in Alzheimer's disease research and caregiver strategies. Thanks for joining us. Welcome back to Dementia Matters. Brain fog is one of the most commonly reported symptoms following COVID-19, impacting an individual's thinking, memory, and concentration. While not much is understood about how brain fog develops, a new study has found that COVID-19 can lead to a buildup of beta amyloid and other peptides related to Alzheimer's disease in the central nervous system, giving new insights into targets for treating brain fog as well as understanding how Alzheimer's disease develops. Joining us today to discuss this very important study is lead author Dr. Sean Miller, a research scientist in the Department of Ophthalmology and Visual Science at Yale University School of Medicine. Dr. Miller, welcome to Dementia Matters.
Thank you so much for having me.
So before we get started, can you share a bit about how you got into ophthalmology and what led you to study the interconnections between the eyes and age-related disorders like Alzheimer's disease?
That's a fantastic question. It all started initially because I wanted to look at early disease detection. So back in undergraduate school and then also during my postgraduate doctoral program and postdoctorate program, I was really fascinated at designing therapies targeting Alzheimer's disease, specifically amyloid beta. However, the issue with a lot of these amyloid beta targeting or even neuroinflammatory drugs and therapies is its inability to essentially prevent disease onset. We're already targeting areas in which we have accumulations of amyloid beta, potentially hyperphosphorylation of tau, and other properties associated with Alzheimer's disease. If we're able to intervene early with therapies, which means we have to detect disease early, we could actually treat before neurons die, potentially preventing cognitive decline, memory loss, and some of these other clinical symptoms associated with Alzheimer's disease. So what had led me to look at the correlation was early disease detection. In 2023, a really hallmark paper came out in Acta Neuropathologica that showed the proteome of the human retina in Alzheimer's disease patients. Really groundbreaking at this time, where it really showed concretely that there's definitely pathology in Alzheimer's disease patients, similar pathology that's conserved with the brain, such as the accumulation of amyloid beta or amyloid beta plaques. This presented me with an idea in which we see the accumulation and pathology in the Alzheimer's disease eyes. This correlates to the human brain. So can we use the human eye, which is a clinically accessible, non-invasive area, in order for us to screen amyloid beta? So in our Science Advances recent manuscript, we also took human retinas from rapid autopsies from Alzheimer's disease patients post-mortem, kept them alive in long-term culturing conditions, and further established, showing with our small molecule crenat-28, which is a curcumin derivative that binds amyloid beta and fluoresces, that we could also indeed detect it ex vivo. So this further confirmed and validated the proteomic findings, and also allowed us an in vivo or ex vivo marker such as crenat-28 to be able to visualize amyloid beta plaques in the retina.
So, Sean, it does sound like, you've been interested from the very beginning in this idea, early detection, because that would lead to early intervention. If you could detect early enough, you're talking about prevention even. And then it came to the eye, though. And it sounds like you inherently didn't just gravitate to the eye, but that's really through your training and research and other publications, you came to this realization, oh, we could do this through the eye, through detecting proteins in the eye. Does that seem fair?
Absolutely. Yeah, that's absolutely kind of how the story evolved. And I want to say is that it's kind of an out of the box idea to look at the human retina and the human eyes to study a brain disorder such as Alzheimer's disease. But when I was initially thinking about it, there was a study that came out from Yale, who's a senior author, Dr. Brian Hafler, that showed conserved or phenotypes between microglia in the Alzheimer's disease brain and the Alzheimer's disease retina from sequencing data. So I pitched the idea to a world-leading retina specialist, Dr. Brian Hafler, the senior author on our COVID manuscript in Science Advances, to essentially look at Alzheimer's disease pathology in the human retina using real clinical samples from patients themselves, postmortem, and also we'll get in further about a real long COVID clinical screening we're performing at the Yale Eye Center.
Oh, that's a really good teaser, Sean. So before we get to that, though, if you could share with our audience, what exactly is this link, or provide an overview of the connection between our eyes, vision, and neurodegenerative processes like Alzheimer's disease, dementia. Well, what has the field in general shown? Because I do think you're right. Our listeners are initially thinking, well, we're talking about brain diseases here, and now you're talking about the eyes and vision. So what is that connection?
So the association with the eye and the brain is really because the eye being a component of the central nervous system is comprised of neurons, different types. We have microglia, we have astrocytes or macroglia-like cells, and we have a blood-retinal barrier. All very similar properties to what we see in the brain, where we see these mixed cell types, but also the blood-brain barrier. The difference being is that with the eye, instead of performing, say, autopsies or biopsies of tissues, we can actually just visualize the amount of pathology in the human retina just by non-invasive clinical imaging, which could potentially have hopes to screen large populations of people just in order to detect the amount of amyloid beta that would be in the central nervous system.
And Sean, there is some evidence too, right? And I say this because we've had experts in audiology come on and talk about hearing loss and the connection to cognition. But there's also building evidence for visual loss, or issues with vision and cognitive change. And does that relate to what you're talking about? The similar cells, the blood brain barrier, the retinal brain barrier, or retinal blood barrier? I mean, is that what you're seeing too in the field?
So we can actually look at the eyes of Alzheimer's disease patients, see what we call gliosis. So this is really the term in which we have inflammation in the retina, very similar to what we get in the human brain of Alzheimer's disease patients. Additional impairments have actually been found with neurodegenerative disorders is actually with night vision. So your impairment of actually dark and light responses in your photoreceptors, for example, have been shown to be altered, particularly in patients such as Parkinson's disease, but we've also been starting to evaluate with our Yale Eye Center is the differences in Alzheimer's disease patients between clinical and numerical data as well, which I'm sure we'll discuss at the end.
All right. So then with that context, Sean, what led you to study the retinal tissue to understand these processes with proteins surrounding COVID in the brain? So when did you make that step to the COVID-19 infection?
So for this was really focused at the antimicrobial peptide response. So again, going back to early disease detection, really kind of puts you around etiology. So you're really just trying to figure out development or progression of Alzheimer's disease. And so what I wanted to do was to further validate the human retina as a model of Alzheimer's disease. First, we showed and characterized that we could visualize plaques. Second, to actually understand now we have a good Alzheimer's disease ex vivo and in vitro model because we show it in retinal organoids and in post-mortem Alzheimer's disease tissue. So we have a good model in order for us to study and evaluate translational Alzheimer's disease. So next we want to say, okay, now what is the effect of antimicrobial peptides or amyloid beta against SARS-CoV-2? We know that there's been papers, especially initially when the pandemic first started, around 2020 and 2021, where a lot of milestone papers came out showing infiltration of SARS-CoV-2 into the central nervous system. They showed in choroid plexus, they showed in the cortex, and they, of course, showed and confirmed it in the lung, which is as expected, which is really fascinating for us to say, okay, now we have not only a good model study of Alzheimer's, we're aware of the amyloid beta being an antimicrobial peptide and having that response against pathogens in the central nervous system. We know that SARS-CoV-2 or the virus that causes COVID-19 can infiltrate into the central nervous system. And lastly, we know that over 750 million cases have been confirmed with COVID-19. And we know that roughly even around 10% of patients can develop long COVID-like symptoms, which I'm sure we'll get to as well, such as related to brain fog.
So it just seemed like a ripe opportunity with the right numbers, the right connection, and then what you're studying with early detection in the eyes. And so before I ask my next question, Sean, our listeners are familiar with the amyloid cascade hypothesis. And so, I mean, we talk a lot on this podcast about amyloid being one of the first identifiable proteins in this process, time going by leading to the development of neurofibrillary tangles, tau protein within the neuronal cell or the brain cell, tau being very closely linked to neurodegenerative processes or atrophy, early death of neurons, and then eventually having symptoms because of neuronal death and synaptic dysfunction. So that is something that's been pretty well discussed on this podcast. But your study is leaning on something slightly different, and you've mentioned it, this amyloid antimicrobial hypothesis. And so before you talk about the results of your study in the paper, can you explain for our listeners what exactly is this hypothesis and how does it build on this overarching amyloid cascade hypothesis?
Absolutely. That's a fantastic question. I'm happy to discuss that. The antimicrobial peptide hypothesis of amyloid beta is really focused on an innate immune response against microbial pathogens. So, your neurons, your astrocytes, and other brain cells will actually secrete amyloid beta in response to pathogenic infections in the central nervous system in a way to suppress the spread of these pathogens. And what we actually think with antimicrobial peptide is that these peptides that form oligomers and fibrils and plaques eventually actually fibrilize and actually entangle or cause a mesh-like netting around pathogens in order to prevent their spread, or what we would call amyloid beta encapsulating these pathogens, again, as an innate immune response in the central nervous system. So, and then with the antimicrobial peptide hypothesis, it's starting to evolve even further. Where now we're showing with viral flare-ups such as herpes simplex virus, which has been largely led in a very cutting-edge study a few years prior, but also now with SARS-CoV-2, we know chlamydia, no fungal infections, in which we see that patients actually get repeated infections over their lifetime. So we think about these repeated pathogenic infections kind of on a continuum where we form an innate immune response, such as the encapsulation with amyloid beta around these pathogens. So the idea being is that you have these repeated, what we would call waves or wave-like features in which you have high accumulations of amyloid beta during a flare-up or during an infection that enters or infiltrates the central nervous system. Then we believe that you get some residual clearance, such as microglia or some non-enzymatic and enzymatic processes. But overall, you have a residual and trajectory of amyloid beta that accumulates with aging. So we believe that the amyloid beta that we see, even in regular aging, that patients get a little bit of amyloid beta, and without cognitive impairment, and of course we see that's exacerbated with patients with cognitive impairment, nonetheless is that we see that with aging it increases. So this has kind of led to what we call the amyloid beta wave hypothesis of Alzheimer's disease.
And so, I mean, Sean, that was a great answer. And I think for our listeners who might not be as technically versed, I mean, you're speaking a lot to inflammation. So infections being present, driving inflammation, but amyloid beta in this context, not necessarily being a bad thing, but actually a part of our body's response to the presence of an infection, part of like this inflammatory fight against infections, encapsulating it and being present. And so, and I think this is really important. Why I'm so glad to have you on this podcast and this show today, because so often people just assume, well, amyloid is bad. Amyloid is not, it's the problem for Alzheimer's disease. But what you're really showing and arguing is, well, in certain contexts, actually amyloid is meant to fight infections and be good for us. But there comes this point where maybe there's too much, or these waves, or perhaps, and please correct me if I'm wrong, but there comes this tipping point where there's too much of it, and then that's what leads to this cascade that I already mentioned, leading to tau. Am I saying that correctly from your experience?
Absolutely, absolutely, you're spot on. Yep, that's correct.
And so, I mean, what I'm really excited to have you on the show is, we can marry some of these ideas of the amyloid cascade and some of these other ideas of infection. I'm so glad you brought up herpes and chlamydia because actually that was one of my later questions for you of other infections. Because sometimes people think these are antagonizing ideas of, oh no, Alzheimer's is not amyloid, it's actually herpes infection. Or I didn't hear you mention the oral hygiene. There's certain bacteria in the mouth. Like they say it as if they have to be separate things. But what you're actually discussing here is, well, no, they are connected. These infections drive this buildup of amyloid, not necessarily to be a bad thing, but it just becomes a bad thing later on. And is that what you saw in your study that came out?
Yes, we saw even that if you would treat with SARS-CoV-2 spike 1 protein, you get an immediate flare-up or release of amyloid beta, which then over time, unless your repeated infection, starts to dampen off. So it's almost like those waves actually are existent in which we're seeing a peak of amyloid and then kind of a settling down after that infection has been essentially immobilized, if you would say it that way.
So then how do you connect this whole process, the COVID infection, the waves or and these acute spikes in amyloid to then brain fog? And in that answer, I'm hoping please define for us how you in the study defined brain fog, how that might be different than other terms that we might use like, oh, cognitive decline or cognitive impairment. How do you put all of that together?
So initially is that the idea with the connection between, say, brain fog, which is really a subjective term, right? It's a non-medical term that we've kind of evolved since really the pandemic has really led to us, considering brain fog or again this non-medical subjective term. So how we actually defined the brain fog for our study was really based on serological tests that were positive
And
for
what we found too
SARS-CoV-2
was,
or
again,
COVID,
these are
and what we would consider a
non-dementia.
severe
So there
infection
was
in the
no
cognitive
clinical
impairment
reports.
prior to the SARS-CoV-2 infection. The patients had died with severe COVID with a positive test, and we confirmed that they had been evaluated for what they would write in their reports as cognitive impairment. So there would be what we would call our, again, subjective terminology, which really just means cloudiness or say like an internal fuzziness in your brain when you're trying to think, such as memory or even different things of, forgetfulness or difficulty multitasking. But and that says brain fog, and which a lot of times are post infection. So you would think of post a severe infection or whatnot, such as COVID. You get this brain fog. Brain fog compared to cognitive decline is a little bit different. Cognitive decline is actually is a medical term in which actually is associated with a decline over aging, in which your ability for memory, cognition, even stuff such as difficulty with familiar tasks or reasoning, and sometimes you'll start to notice clinical symptoms such as, say, deficits in communication as well. So that's kind of like where we show the difference between cognitive decline and brain fog. So then where do we think that, okay, we see amyloid beta in COVID-19 patients that had severe infection upon, say, death. And then we're looking at their eye, which we know has amyloid beta that's associated with Alzheimer's. How does that connect now to the brain and brain fog? It's a very fascinating question, and this is kind of what had led in now to a new approach that where we hopefully will have published very soon, is that we're actually looking to see on an electrophysiological level around these plaques what is actually happening with the neurocircuitry. Do we see changes in the amount of, say, potassium signaling or calcium signaling in specific neuronal populations that are only around plaques? If that's the case, we can actually do electrophysiological recordings in the retina to establish to see, do we see changes in the retina on this level that may be associated with what we see in the brain. And how the connection actually has is we're using these rapid autopsies to do electrophysiology around the plaques in these ex vivo, still viable cultures. So that's kind of how. Okay, so we are thinking more that these plaques are causing a miscommunication or communication changes within the neurons around those plaques. So that's kind of where this has led. We're now at Yale Eye Center. We are running with the long COVID clinic here at Yale retinal imaging for patients who have brain fog, who have had HIV, and who have also been our controls, are actually no cognitive impairment, age matched controls. And we're finding patients as young as even in the 40s with some what we would call proteinopathy that are associated with brain fog. And it's really fascinating. So maybe we can make the connection between amyloid in the eye and amyloid or cognitive dysfunction in the brain. And one I just want to end on is that in addition to the Yale Eye Center that we're evaluating, is we're actually establishing artificial based tools such as machine learning that goes and does the detection of patterns or fluorescence, or what we would actually look at is hypopigmentation in the retina and for proteinopathy on just a regular eye exam. And we can find within about 10 seconds, you can actually screen about 40 different patients who have their what we call fundus autofluorescence microscopy with our artificial based model in order to say yes or no with a disease classifier. Are we detecting abnormal protein deposition versus, say, normal with aging? And again, this is with establishment with the Long COVID Clinic and Yale Eye Center.
That's incredible, Sean. And I mean, it's always exciting when we talk about artificial intelligence, AI, machine learning. But I mean, you're really using lots of rich data and help and getting the assistance from machine learning to do it. But it also, what you're speaking to shows the true benefit of these rapid autopsies, because I wasn't really fully grasping it until that answer of, these were really generous individuals who were dying, knew they were about to die, consented to being having this done. And so that allowed for you to have really living tissue post their death, but living tissue that you could then study. And you mentioned in your answer, not only, so you have this incredible resource thanks to this unbelievable donation, but now you're also looking at HIV. So it does go to my question that I wanted to ask earlier of, is there something unique to viruses or these other bacteria that you mentioned chlamydia, and then I mentioned the oral dental? Is there something that where it has to be a head and neck infection? Or, I mean, what makes you think certain organisms are triggers versus others? Do we have a theory behind that?
That's a great question. To be honest, when it comes to specificity of, say, amyloid beta versus these pathogens, I just don't think the literature is enough out there to really establish, is it ubiquitous or is it a common mechanism that happens, or is it specific to certain viruses or pathogens such as gingivitis or some of these other ones as well. And one thing I want to mention though is that these more severe infections cause systemic inflammation. And I think we'll probably end up touching on it is more in which the systemic inflammation also impairs the blood-brain barrier and also the blood-retinal barrier, which makes it more permeable. So it allows more infiltration of pathogens with aging, because in aging we have a natural lost integrity of the blood-brain barrier, but in Alzheimer's is exacerbated. And this is actually, and in high risk populations, so the systemic inflammation that is actually worsening even your blood brain barrier, which is allowing even more infiltrates of these pathogens into the central nervous system.
No, that actually, and that, Sean, that was my next question about the blood-brain barrier, because I read that in your paper. So that seems to be a very critical part of this idea of infection, because some infections can actually just be in the brain, right? So they identify the virus or the bacteria on autopsy. But what you're saying too is other infections create whole body inflammation, loosens up sort of the blood-brain barrier, which allows for more infections to get in, or at least inflammatory proteins. Am I saying that correctly?
Absolutely. That's exactly at least what the thought of mine has been going. And I think some of the research is leading to in the field.
Well, one, it shows the importance of the integrity of our blood-brain barrier. You've given me an idea for another podcast, so thank you, Sean. But I also read in your paper you mentioned this target, so amyloid aggregation being reversed in your study when you targeted an anti-tumor site. So we've been talking about infections and inflammation, but then your paper also talks about anti-tumor sites. So what is the connection with this?
Fantastic question, and really excited to actually get into this one. Is that so? How we actually first started to look at it was we did single cell RNA sequencing from postmortem Alzheimer's disease. Again, these rapid autopsies, patients from their retinas. We compared it to age matched controls as well, and we also have done COVID. And what we found was that instead of being ACE2, which is a common receptor that people think about when they think about the SARS-CoV-2, we actually found another receptor especially on neurons called neuropilin-1, which has been, as you mentioned, associated with tumors. So with this is that we found that through spike 1 protein, this is the mechanism in which we're able to elucidate, is that it's binding to neuropilin-1 and then increasing amyloidogenic pathways. There was a paper that potentially showed it working through gamma secretase. So there's some really fascinating research coming out recently kind of surrounding this topic. But we did find that if you block this interaction with neuropilin-1 blockers or inhibitors, at the same time you're exposed to SARS-CoV-2 spike 1 protein, we found that there was a lack of accumulation of amyloid beta, suggestive that the MOA or mechanism of action would actually be this neuropilin-1 receptor, since we don't see ACE2 on neurons, And there's a beautiful paper that actually had showed that neurons can be infected with SARS-CoV-2 through neuropin-1. It was a beautiful science article.
I see. So now we're getting a level deeper, and you're saying, well, we see this relationship with COVID-19 and amyloid, but then you went further, you're able to show, well, now we can see how this is happening, and it's through this receptor that deals with tumors.
Yep, absolutely. And I just want to add is that listeners out there will think, okay, well, how are you supposed to be treated with a neuropilin-1 inhibitor the second that you get SARS-CoV-2 when you may not know you have SARS-CoV-2 until testing, for example. And unfortunately, nowadays is less frequent for people to test themselves for COVID-19 as it was during the peak of the pandemic. But nonetheless, is that if we're able to really, instead of doing a treatment at the same time that you're exposed to SARS-CoV-2 spike, one protein or SARS-CoV-2 infection, is really to be able to treat it a few days after. So the idea being is instead of an antiviral drug, if you think of it that way, is actually be an anti-amyloid drug or an amyloid enhancing drug for clearance, where you'd actually clear out excessive amyloid or reduce the amount of amyloid that's being produced. So the idea would be that maybe it works as an antiviral, or maybe it's just an anti-amyloid, but it could have both mechanisms, or it could work both ways even. So I think that that's a better approach than trying to target initially, which shows great MOA, right? That was the goal, was to show mechanism of action was clean. We understood it was through that from single RNA sequencing. And one thing, and a very fascinating thing, I'm sorry that I didn't mention, for our drug screening, and that this was in the article, is that we actually made human retinal organoids. So we took blood draws from patients, and these are controls and also from Alzheimer's disease, and we can make them into induced pluripotent stem cells and then differentiate and mature them into really human retinal organoids that are multicellular and recapitulate the cell types in the retina of the human. We then use high throughput screening. So we did drug screening with different neuropilin-1 blockers. And this was using, again, that crenad-28 or curcumin fluorescent derivative that binds to amyloid beta 42, in which we would essentially just add spike 1 protein, add this crenad, see increases in fluorescence in green, for example, with crenad-28, which targets amyloid, and then just apply a lot of drugs to essentially see where do we get rid of these plaques. And we found that an optimal neuropilin-1, which we had published in the paper then, that worked really well.
No, thank you for explaining that process. And I just think that even for those of us that don't fully understand everything you just said, we can appreciate that this is what leads to eventual new targets, new ways of treating, disease. And especially since you're showing this, antimicrobial amyloid connection. Well, it's not just for knowledge sake. There is actual opportunity then for drug treatment. And Sean, that kind of leads to my last question today, which is really, it seems like there's lots of next potential steps, but where is your research in particular going from here? Like, what are you going to be continuing to focus on this antimicrobial hypothesis, or are you going to move into clinical trials? Are you going to do different drug development? I mean, where do you see the next few years?
So it's really the future is pretty exciting. I would say, at least in that way, is that not only do we have the ability now to look at the antimicrobial peptide response in our human models, but we can also now start doing drug screenings, rapid drug screenings with even more additional targets with our organoid model. And we're currently having that go on right now with high content imaging. Not only will we find additional drug candidates and targets with our high throughput organoid screening, we not only have that, and those candidates then will hopefully lead to clinical trials. But in parallel, to understand, can we even use the retina to further understand in the clinic? Again, I mentioned a little bit earlier, is that in collaboration with the Long COVID Clinic at Yale, and then with Dr. Brian Hafler, who's a senior author, and myself as lead author on the paper, we now are working with the Yale Eye Center to do clinical screening for proteinopathies in the retina of real patients. So this is ongoing, and we're still, of course, recruiting. So, always happy to help anyone or answer questions regarding that. So that's our goal, is like, can we get crenad-28 to clinic so that we can look specifically at amyloid beta in the retina? So that's one step. In parallel, we're also working again with the artificial intelligence-based tools to do patterns and to recognize into our disease classifier, when do you reach a threshold that's essentially what we would consider too much amyloid. And then addition to that, would be that we're still trying to figure out this antimicrobial wave hypothesis or amyloid beta wave hypothesis of Alzheimer's disease. So drug candidates from our high throughput screening, Yale clinical screening from proteinopathies, and trying to get something specific for amyloid beta so we can distinguish between other proteins or even lipids and amyloid beta when we do our just regular eye exams that take just a few minutes.
Well, that's all very exciting, Sean. And for our listeners who are in the Yale area, I'm certainly happy for this podcast to be a recruitment source for you. And so if you're interested, it sounds like you should contact Dr. Sean Miller. Very exciting trials or studies happening. And I certainly look forward to those next publications, Sean. But let's end here today. And I want to thank you for being on the show. And we certainly are excited about the future papers coming.
Appreciate it so much. And this was fabulous conversation. Really appreciate it.
Thank you for listening to Dementia Matters. Follow us on Apple Podcasts, Spotify, or wherever you listen. Or tell your smart speaker to play the Dementia Matters podcast. Please rate us on your favorite podcast app. It helps other people find our show and lets us know how we're doing. If you enjoy our show and want to support our work, consider making a gift to the Dementia Matters Fund through the UW Initiative to End Alzheimer's. All donations go toward outreach and production. Donate at the link in the description. Dementia Matters is brought to you by the Wisconsin Alzheimer's Disease Research Center at the University of Wisconsin-Madison. It receives funding from private, university, state, and national sources, including a grant from the National Institutes on Aging for Alzheimer's Disease Research. This episode of Dementia Matters was produced by Caitlin Rowerdink and edited by Alexia Spevacek. Our musical jingle is Cases to Rest by Blue Dot Sessions. To learn more about the Wisconsin Alzheimer's Disease Research Center, check out our website at adrc.wisc.edu. That's adrc.wisc.edu. And follow us on Facebook and Twitter. If you have any questions or comments, email us at dementiamatters@medicine.wisc.edu. Thanks for listening.